This unit starts off with a brief introduction to just some vocabulary terms, all right? And the ones that you need are genes, chromosomes, DNA, things like that. So now, chromosomes you probably already know. So chromosomes are stretches of DNA there that are present in the nucleus of a cell. And you should know how many humans have, okay? So in human body cells, which basically means all cells other than gametes, we have a total of 46 chromosomes, all right? In gametes, we have half that number, so there are 23. Now, that means that when you were a gamete, okay, you weren't a gamete, when you were a zygote, it means that you got 23 from your mother's gamete and 23 from your father's gamete, which is why you now have 46, all right? And this is why you have meiosis in terms of the division of cells, chromosomes in half, all right? That's pretty straightforward. Genes has a straight up definition. You should just have it memorized. It's not a difficult one, but basically a gene is a stretch of DNA that codes for a particular protein, all right? And if you want to know how that happens, you need to go and watch the video Protein Synthesis, where I talk about transcription and translation and how all of that stuff works together. Sorry, I've lost my camera. I need to make sure this is in focus. There we go. All right. Stretch of DNA that codes for a specific or particular protein. Okay, you have about 25,000 genes in your genome, and a lot of your DNA actually doesn't code for anything. It's doesn't, or it's not, it doesn't code for genes. All right. So, and if the structure of the DNA, I talk about that in another video as well, where I talk about RNA versus DNA. Okay. And the structure of DNA as a double helix. I talk about the RNA nucleotides, things like that. All right. What I don't talk about in those videos, so I'll talk about here, are mutations, all right? So the idea is a gene is a stretch of DNA that codes for a specific protein. Okay, check. Then all genes can have mutations introduced, all right? And so a mutation is simply a change in the sequence of bases in the DNA. And the idea is that that change in the sequence of bases can then result in a change in the amino acids that are present in the protein that it's coding for. Right? There are 4 types of mutations that you need to know. The first one is a duplication. Okay. Then you have deletion, substitution, and inversion. I'm going to get you a stretch of DNA, and we're going to talk about what these 4 look like. Okay, so here I have a stretch of DNA. These are the bases on the DNA. Okay, I put them in triplet codons just so that it's easier to see what's going on. All right, and a duplication is exactly what it sounds like. It often occurs when you have a stretch of similar letters all in a row. Right, and so the idea is that if the DNA is replicating, it would come along and it would say, okay, there's an A, T. It would duplicate that, and then the C. Oops, it's supposed to be a C. But then here in the GG, it might say 1G, another G, and then it would duplicate this base and put a G here instead. And then it would continue on its merry way. So what you've done is gone from 2 Gs to 3, all right? But then it would still continue with the rest of them. So it would have a G, A, T, C, C, T, T, A, G, and a G, like that. All right, so this is a duplication. Now, the problem with this, of course, is what you can see is because I've kept it in the 3s, is that it causes what's called a frame shift, which means that the AAT and the CGG are correct, but then after that, now instead of ATC, you've got GAT. Instead of CTT, you've got CCT. Instead of AGG, you now have TAG. So it causes a frame shift, which means that all of the downstream codons are now completely different, all right? So it causes a frame shift and affects all downstream codons. So these can be really, really deleterious to the organism, all right, if they happen in certain areas. Okay, so that's a duplication. The next one is a deletion, and a deletion also causes a frame shift. So it has the same, it's a different type of mutation, but it still has the same thing, all right? So let's say, I'll stick with this G here, okay? So now I have AAT, that's fine, C, G, but then if it deletes the G, all right, instead, I'll write down loss of G, then it'll skip over the G and it'll go directly to the A here. So this will be A here, CGA, two, right? Instead of having two G's, it has one G now. And then it'll, hey, and then this will be a TCC, right? TCC, and then a TTA and a GG, and it would continue on. Okay, so this also, the idea is that if you look, right, the AAT again is fine, but the CGG now becomes CGA because you've lost a G. And then again, all of these now look nothing like their downstream counterparts or what they should be. So both of these cause frame shifts, and they can be really deleterious, or be really bad news, lead to a non-functioning protein. All right, the substitution is also often called a point mutation, and it's not nearly as bad. Overall. It can still lead to a non-functional protein, especially if you're looking at an enzyme or something like that in a certain area. A substitution is where one base gets replaced for another. So you would still have AAT, CG, and then if we're talking about the second G, maybe instead you would get a, I don't know, a T put in here instead of this G, and then it would continue on ATC, CTT, AGG. And so what you can see is that this codon is different, right? CGT instead of CGG, but all of the others are still the same. So it only affects a single codon. It's called a point mutation, right? And then the last type of mutation is an inversion. And an inversion is where one of the codons basically gets flipped. So let me write that out. So you'd have AAT still, and then you'd have GGC instead of CGG. ATC, CTT, AGG. All right. Now this, the GGC, right? This has been inverted, but you'll notice it still only affects one codon, right? The following ones are still the same. So this changes one base. This changes one codon, which can still be bad, but again, it doesn't cause this frame shift in which every single downstream codon is different. All right, so those are your types of mutations, and then of course you need to know what can cause a mutation, and those are called mutagens. So mutagens is anything that increases the rate of mutation. So examples of this would include, I've lost my video, can you see this? Sorry, this would include any sort of gamma rays or X-rays, UV light, lots of different toxins or chemicals can cause them. So X-rays, gamma rays, UV, lots. If you're in physics, you know about a lot of these. Many different chemicals can cause. And so, the thing is about mutations, right, is as hopefully evidenced by this, is that most mutations are bad news for an organism. All right, they have a detrimental effect on organisms. So most mutations are detrimental. Some of them have no effect at all. All right, so I don't know, for example, the blue eye, that's a mutation that occurred randomly in humans many, many generations ago, and there's no detrimental effect to that. It just is different, right? Having blue eyes versus brown eyes doesn't mean that you're less or more likely to survive. It's just a difference. So most mutations are detrimental. Some, I guess I'll say, are neutral, and very few, but some of them can be beneficial. So an example of this would be, for example, via random mutation, a bacteria might be resistant to an antibiotic. Well, that's a great thing. That's an advantage. Well, for the bacteria, it's a great thing. It's an advantage to the bacteria if, for example, that antibiotic is around. It will survive and all of its fellow bacteria that don't have that mutation will die. Okay, so most mutations, however, are detrimental, some are neutral, and very few of them are beneficial. And the idea of accumulated mutations, this is how evolution happens, right? When we talk about natural selection, we talk about mutations, all right? And you don't get to pick and choose your mutations. Mutations are random, right? They are random changes that occur randomly, random throughout the DNA.